Hi everyone. We can, hello. If we can take our seats back, please. Thank you. Firstly, thank you for your patience this afternoon. I will give a very, very brief introduction because I'm sure you're all much more keen to hear from our keynote speaker this morning, Professor David Nutt. So, very briefly, I think we all know he's had a long, illustrious career in research and is currently the director of, see if I can say this correctly, neuropsychopharmacology unit at Imperial College London. But perhaps more widely, we know that his work and his experience straddles some of the key areas and themes of our discussion today. Realizing the opportunities and addressing the challenges in research and innovation really requires synergy, engagement with academic research, but also in innovation and policy. And it's this engagement in policy that I think we all find particularly interesting. In addition to his position on numerous advisory boards and councils, both within the UK, the EU, and globally, he has been a vocal advocate for the role of research in addressing challenges, societal challenges, and particularly the importance of evidence-based policy making. And with, I think, that in mind, we know that he's been engaging proactively in all the strands of the quadruple helix, in addition to his public engagement work, and through this work has been able to enact meaningful change in the area of harm reduction. So I will now please welcome Professor David Nutt. Thank you.
Well, it's can you hear me all right? Yeah?
Yeah.
It's very good to be here. Thanks for the invite. If you told me you had a drinks reception afterwards, I'd have provided the alternative that I'm going to talk about, but just remember that for the next time. And if you want to read more, there's some books. Yeah, I gather that you're the future of leadership. I suppose probably you invited me as an example of a failed past leader. That's me being sacked as the chair of the Advisory Council on the Misuse of Drugs. The hand over my mouth, anyone know whose hand that is? He was a Home Secretary at the time. Obviously, actually a long way from here, the other side he was in from Hull. He's Alan Johnson. He was trying to shut me up because I was saying the British drug policy wasn't evidence-based. I was saying that cannabis was a medicine. And on the bottom left-hand side of the image, you can see the scales of justice tipped in favour of alcohol and tobacco, which are clearly the most harmful drugs in the UK. And the hysteria that was being created before the 2010 election around mephedrone, MCAT, meow meow, was just a smokescreen to avoid people confronting the truth of the drugs which cause most harm in society. That conflicted with government opinion at the time, and so they got rid of me. Thankfully, they didn't shut me up because it's quite difficult to do. Many people have tried. And there's an article in the Times, sacked advisor warns of alcohol time bomb, and well, that is what I'm going to talk about today, how we can avoid the alcohol time bomb by using science. As I was sacked, a more educated colleague of mine wrote to me this little note. David, he said, you obviously haven't read Voltaire because you would know it's dangerous to be right when the government is wrong, which is a general principle that you're all going to have to live with if you're going to change things. So why did I say that alcohol was the most dangerous and problematic drug, certainly more problematic than the ones that are controlled at the time? And that's because of this analysis here, which we published in the Lancet a couple of years later, the most sophisticated analysis of the harms of drugs that's been done using a technique called multi-criteria decision analysis. And I can suggest to you all, if you have to engage in decision-making, complicated decision-making, where there are many variables with different dimensions, you should do MCDA. It is unquestionably the most powerful way of disentangling both problems and solutions. It's not easy to do, but it's well worth doing it. Not least because if you do it, you learn an awful lot about what you don't know. That's another story, but I highly recommend any of you to think about using it because it is a very powerful technique. And having done that, having spent several conferences with groups of experts doing MCDA on drug harms in the UK, we came to this conclusion, which is on the left-hand side. You see the size of the histogram is the magnitude of the harm of each different drug to society, to this country. The red bars are the harms to society. The blue bars are the magnitude of harms to the individual. And you can see that alcohol is the most harmful. And they scale down to, on the right-hand side, magic mushrooms are the least harmful. So that's exactly the opposite of what the current government drug policy says. And it's wrong, therefore. However, that wasn't just a UK analysis. It was also, it's been replicated by the European Department of Justice through 30 different European experts, 20 different countries. It's been replicated in Australia. It's been replicated in New Zealand and replicated in a paper that's in press now in Canada. In all Western societies where alcohol is a legal drug, it is the most harmful drug. And that is because of the harm to other people, to society. It's not the most harmful drug to the user, which is reassuring to you because most of you are going to drink tonight. But overall, alcohol is the most damaging drug. And it's the leading cause of death in men under the age of 50 in this country, and it will be the leading cause of death in women. It may already have achieved that status, but they're holding back the data because I think they're terrified to discover that that is also true. The last data we had just before COVID-19 was a 6% rise in alcohol-related female deaths in 1 year, which is absolutely unprecedented rise in deaths from almost any drug other than fentanyl. So it's a big problem. It kills a lot of people, particularly young and middle-aged people. Why does alcohol exist? Well, why are you going to drink tonight? Well, you're going to drink tonight because it's going to make your evening more sociable. In fact, it'd probably be pretty miserable if you didn't drink tonight. Oh, that's just a thought. I mean, it's a hypothesis. You can test it if you want. So alcohol exists because of its pleasurable effects, and particularly the effects that alcohol has to bring people together. And it's something I guess almost all of you have experienced. It encourages togetherness, it encourages us to socialise, it encourages us to get intimate with people, and it does it through changing the brain. And over the years it's become evident that one of the main targets, almost certainly the major necessary target of alcohol in the brain to produce the effect we want, which is relaxation, sociability, conviviality, is through activating a neurotransmitter in the brain called Gaba. Gaba is the main inhibitory neurotransmitter in the brain. It calms the brain down, which is what alcohol does. The first thing they do on aeroplanes when they take off the seatbelt sign is to serve alcohol is because so many people in the air are terrified, and alcohol is a relaxant. And there's an interesting debate going on between anthropologists about why human society actually ended up being as successful as it is. And the success of humans comes from the fact we stopped being nomads and we started settling down in villages and then towns. And the drive to settle down was because we learnt how to cultivate cereals. And when you've got to wait for the cereals to grow, and while you're waiting for them to grow, you can do other things like develop language and architecture, writing and maths, etc. And you hopefully all read this book by Johan Hariri, A Brief History of Humankind, Sapiens. He argues that it was actually the planting of wheat to make bread that made humanity. So wheat eventually actually civilized the human species. But there's another argument which I think is slightly more compelling, and that's Edward Slingerland, who argues that we grew wheat in order to make beer, and we cultivated obviously grapes to make wine. And that is arguably a more compelling argument, not least because, as most of you will probably have accidentally happened upon sometime in your life, being drunk is a very good way of making friends. And making friends with people you don't know is often a good way of spreading the gene pool. And that is why he believes that places like Stonehenge were built to encourage the coming together of different tribes. Because, as I guess most of you know, there was a period when Homo sapiens had we were small in number, and interbreeding could have created serious problems. So we wanted to outbreed, and alcohol is a very good outbreeding medium. But of course it also comes with problems. I've talked about the deaths, but also it's strongly associated with other harms, particularly accidents, violence, etc. And a lot of people, maybe about 15% of people who drink, become addicted. And that is a serious problem because we don't really know how to stop that and how to help them when they are addicted. But most importantly for the talk today, it's a really old technology. In fact, I would argue it is the only technology we use today that was founded 40,000 years ago when probably sapiens in the Rift Valley of Africa learnt that you could ferment honey to make mead. That was probably the first drink. Of course, they were picking up rotting fruit long before that. So why do we rely on a technology that is that old? And the answer is because no one's really thought about doing anything different. Or the ones who have thought about it have not managed to convince other people that that's the right way forward. But it's changing now, and the origins of my interest in this came from this government report from the Foresight programme in 2004. That was a very interesting experience for me. I was the medical lead of that programme, looking at the impact of neuroscience on drugs and over the next 25 years. We're getting quite close to the 25th year anniversary of that book now, and most of what we suggested actually hasn't happened, but I am working on it. And in that programme of essentially doing a lot of brainstorming over the course of a year with various experts in various aspects of science and pharmacology and sociology and anthropology, it became clear that it would never be possible to find an antidote to all the effects of alcohol. It's too promiscuous a drug, and it's too damaging a drug. So we hit on the idea that maybe what we need to do is replace it. And that article in New Scientist came out from that Foresight book. So it makes sense to find a safer alcohol if you can. How can you do that? Well, you can explore how alcohol works, and then you can hopefully create something to do what we want alcohol to do without the harms. So we want to find a molecule or a substance or a cocktail that mimics the positive effects of alcohol, something that doesn't affect all the bodily systems that alcohol affects inevitably when you drink, before even it gets to your brain. And we don't want it metabolized to something as toxic as acetaldehyde, which, as the chemists here know, is only one carbon removed from formaldehyde, and formaldehyde is what we preserve dead bodies in. Now, alcohol is very promiscuous. It targets almost every cell in the body. It targets almost every cell in the brain. This image of the brain is color-coded. Red is bad. There's a lot of red, isn't there? Black is very bad. That's death. And but the green bits are good. And the green bits are impact of alcohol in the frontal cortex, which is the most recently evolved part of the human brain. It's the part of the brain which makes you as scientists and creators and visionaries as clever as you are. But it's also where we think about our relationship with other people, where we get thought loops about not being good enough, not being clever enough, not being attractive enough. And alcohol dissolves those, and it dissolves them to make us sociable and convivial through enhancing the effects of Gaba in those brain regions. So it's pretty straightforward. All you've got to do is just do that, just enhance Gaba in your frontal cortex, and all will be well with your party tonight. But GABA is very important. It's the main off switch of the brain, and therefore it's not a trivial exercise to target switching off GABA just where you want it. Because if you switch it off or switch it on elsewhere in the brain, all sorts of problems will happen. You'll fall asleep, you'll fall over, you'll slur your words, etc. So I just want to give you a very... are any of you neuroscientists? Okay. Well, correct me if I'm wrong then. But the brain is a very simple kind of machine. It's a machine. It's about 80 billion on-off switches, and the on switch is glutamate, and the off switch is GABA, and GABA basically controls the activity of the brain through stopping neurons firing, and everything else is all the other things that you hear about: dopamine, serotonin, noradrenaline. They do lots of monitoring and modulating the effects of these core on-off switches, but so they set a tone, but they don't drive brain function in the same way. So glutamate's the on switch of the brain. Everything you have heard, seen, thought in the course of the last 10 minutes of my talking here, everything has been laid down and analysed, and if you've remembered any of it, laid down through glutamate. Glutamate basically is the on switch, as I've said. But the problem is too much glutamate makes you anxious, and if you have even more glutamate, you have seizures. And so over the last few 100 million years of evolution, every time brains, whether they're mammalian brains or other brains, release glutamate, they almost always release GABA to counteract the effects of glutamate. And your brain is wired so it's impossible just to release glutamate, otherwise you would all be dead while you were babies. And so activating GABA then dampens down the effect of glutamate, and you walk a tightrope. Your brain is continually walking this tightrope between too much excitation, too much inhibition. It's quite challenging to keep it perfectly balanced. And the problem with alcohol is that alcohol potentiates, it enhances the effects of GABA, and it blocks the effects of glutamate. And that's why, if you take too much of it, you die. And that's what happened to Amy Winehouse. And most of us, when we heard she died, we presumed it was some exotic drug, maybe, or maybe just the heroin she was addicted to. But in fact, it was alcohol that killed her, because she was an alcoholic as well as a heroin addict. And she was clean. She stopped taking both. She met the criteria for recovery that the government at the time set, which is 6 weeks of not using a drug. But it was that not using alcohol that killed her, because it was very easy for her to relapse to alcohol. She walked down the street, went to the corner shop, bought a litre of vodka and drank it. And she died of alcohol poisoning because she'd lost tolerance. If she'd actually carried on drinking, she wouldn't have died with the blood alcohol level that killed her, but because she'd lost tolerance. In the same way as heroin addicts lose tolerance and they die when they start using again when they leave prison, she died when she relapsed to alcohol. I've already mentioned this, but I just want to emphasize that there's a huge amount of literature that suggests that enhancing GABA in your brain is quite critical to producing social bonding, which is what most of us want to do. Humans, we are so successful. We are just, we control the globe. Eventually we'll control Mars if people are stupid enough to go there, but we can control anything we want because we work together in enormously powerful teams. So we're very, very successful socially, but we're actually not very good at being sociable. And that's why alcohol still exists, because alcohol allows us to fulfill that social role. It overcomes the social anxiety that most of us have. So a few years ago, I set up this company called Gaba Labs in order to make GABA acting agents that would mimic the effects of alcohol, on the supposition that these would be of interest, they'd be popular, and wise people would start using them because they would like to live longer than as they would if they were drinking alcohol. So that's the plan. It's a small company, but we've been quite successful because we've linked with various academic groups who know a lot about the different aspects of GABA and how you can make molecules or find molecules in nature which will mimic the effects. If you want 2 you can read. That's a free download. There's a paper we wrote a couple of years ago outlining the thinking behind what we do, which is targeting both synthetic molecules and also botanical molecules to mimic the effects of alcohol and avoid the unwanted effects of alcohol. Now, I said at the beginning it's complicated, and it turns out that the complication is the opportunity. So GABA is an enormously important neurotransmitter, and there are at least 15 different subtypes of GABA receptors in the brain, and they're all made up of these 5 separate proteins which come together to form this it's like a bracelet which sits in a cell membrane and allows ions to transfer across the cell membrane, and that is how the brain is calmed, by opening of the hole in the middle of that complex. But those 15 subunits come in different families. There's alphas, there's betas, there's gammas, there's deltas, there's pis, and they are distributed in different parts of the brain. And that allows us to potentially target parts of the brain where ones might be overexpressed or highly expressed, and have a specific action. This is just a cladogram of the evolution of these different receptors, and they show there are some large proportions of them have particular subtypes, but even the ones where you've only got 3 or 4% of the whole total of a particular complex, there are more of those than there are dopamine receptors in the human brain. So GABA is the critical controlling transmitter. So what we need to do is we need to target the subtypes which allow us to relax. And we know from, certainly from rodent studies, that there are very specific localizations of particular alpha receptor subtypes in different parts of the brain. And there are 4 main alpha subtypes. They're listed here. You can see they have very different distributions. Hot is more. I won't go to all the different brain regions, but you can see that they're quite different. And they're particularly of particular interest are the alpha 2 and alpha 3 because they gate the cortex. The cortex is this is a rat brain. The cortex is a lump on the top. So those GABA subtypes gate activity in the cortex. The top one, the alpha 1 subtype, is all through the cortex. And that's why you fall asleep if you take a sedative, or some of you fall asleep if you take alcohol, because you basically wipe out brain function. And that's what anesthetics like propofol do. But you can target the smallest, more rare subtypes without producing sedation. You can produce relaxation. And we're now beginning to use PET scanning to identify these subtypes in different parts of the brain. This is the alpha 5 subtype, which is a lot of relevance to addiction, but not particular relevance to what we are talking about today. But I'm just showing you the technology can be extrapolated to humans. So this process has actually met with quite a lot of interest from cartoonists. I think you kind of know you're on the right track when the Times will make a cartoon about your drink. So this is the building up, I think, to the 2015 election. And it's interesting how historic it is. You see on the left-hand side there, UKIP, unfiltered small beer made from forage. And then you've got Miliband, and you've got George Osborne's Housing Bubbly. You've got Blue Orange Juice, which has got something to do with Nick Clegg, and you've got Mr. Cameron's Absinthe. Anyway, I just thought that was kind of amusing. I wasn't going to invent those drinks, right? But if I did, you would have to drink them very responsibly. But in reality, we've been progressing this in 2 ways. We've been developing botanical drinks that have the effect we want, and we've also been making small molecules that target those particular GABA subtypes and do it in a way which mimics the time course and the efficacy of alcohol. And the botanicals are here now. Some of you may have seen them. They make fantastic Christmas presents, by the way. And as I said, if I'd realised you were going to have a party afterwards, I would have got some sent. but there's always next year. These are botanical spirits. They are basically made up of a host of herbs which are in the food chain, have been in the food chain since before 1997, which means they've been grandfathered in as food safe. They contain some herbs which have molecules which enhance GABA, and they contain other herbs we put in there which have molecules that facilitate the uptake and the penetration of those particular molecules into the body and to the brain. And they have various different flavors, and they can be mixed with any mixer you want to give you a pleasant drink. And we thought, well, that's it, job done. Anyway, we've actually got an alcohol alternative. It works. I can show you. I'm going to show you in a minute that it works. So do we need to do more? And the answer is, it turns out we do, because it turns out most people actually are quite happy drinking what they drink, and they don't really want to explore a different kind of drink that's got a funny Italian name like Sentia and looks different and tastes different. So we've had to revise our thinking as we've gone along, but at least we have achieved the goal. We have it out there, it's available in the UK, in America, and also in Europe. Now the scientists amongst you will say, well, prove it works on GABA. So we started to develop research to address that question. And we began by using mammalian tissue, where we know the effect of alcohol is mediated through GABA, and this is tension in a mouse gut. And you can see on the left-hand side, you can see the gut. As you know, your guts have rhythmic activity, they're pumping away all the time, making food move through it. You put alcohol on, there's a bit of relaxation, the actual magnitude of the excursion goes down, there's a transient reduction in tone, and then it comes back. On the right hand side you see Sentia. Sentia massively reduces tone and produces a prolonged attenuation of tonic activity. And many people describe that. Many people say one of the nice things about drinking Sentia is it actually relaxes your gut. When you lie in bed, many people have that gut tension before they fall asleep, and Sentia eliminates that through GABA. We know that's through GABA because we can reverse a significant proportion of the effect in the gut by blocking GABA receptors. So we're pretty confident we're enhancing GABA in the gut. But that isn't where we're really targeting the effect. We're targeting the effect in the brain. Now, it's difficult to study specific receptor subtypes in whole animal brain. Animal research has virtually died. Many universities have got rid of animal facilities. In fact, the university we started working in, so those gut bath studies I showed you in the previous slide were done in the University of Portsmouth. They no longer have an animal facility. That facility ended last year. In fact, I think on the whole south coast of England now, there's only maybe 2 universities that have actually animal facilities. So it's difficult to do that work. But what you can do, and this is one advantage of modern technology, is you can mimic a mammalian brain, or at least a mammalian receptor. You can actually make a frog or a toad oocyte express a particular receptor subtype you want, and in that oocyte you can then do voltage clamping on the specific receptor subtype. So you can actually then invent molecules or find out whether your molecules work on the subtypes you want to target. And that we've been working with Jerome Swinney in the University of Portsmouth to do that. So this is human RNA expressing specific human receptor subtype in an oocyte. And you can see on the left-hand image there, you put GABA on, the receptor opens, ions flow into the cell. You can measure the change in ion flow with the voltage clamping, and you can show that molecules we've invented enhance that effect. And here is one of our lead molecules. So this is a dose-response curve. The one that's going up is what we want. We want to enhance activity through that particular target. And we don't want to enhance activity through the unwanted target, the blobs. We want to separate the impact of molecules to produce relaxation, but not sedation or ataxia. And we've begun to achieve that with this particular molecule and others that we're developing. So we can make molecules that fit the purpose. What we need to do now is obviously get them into humans. But we can also use those humanized oocytes and asked the question, well, do our botanical drinks do the same? And the answer is yes. The botanical drinks also enhance GABA function in those oocytes. That's the red bar. So there's GABA. You add Sentia, you get a bigger effect. And if you block GABA receptors by cooking it, you reduce the effect. So some of the effect of Sentia, at the least, is mediated through a particular subtype of GABA receptor. Well, what about humans? Well, the good thing is that now Sentia is available and people are buying it. There are scientists that work on it. And this guy, Alex Shaw, from University of Exeter, rang me up a year or so ago and said, did I know that Sentia changes the EG? And I said, oh, wow, how do you know that? And he said, I've done it. And I said, well, that was very kind of you, you know. So I sent him a few more bottles as a reward. And EEG is essentially a measure of electrical activity in the brain, or it's a signature of the different firing rates of different pyramidal cells in the brain. You're all familiar with it, I think. And this is the first data that our drink changes EEG function. And what you see here, if it's red, there's more power in that frequency band, and the frequency bands are stacked from left, slow frequency, to right, high frequency. So there are areas of the brain where there's increased power in a particular frequency, and the alpha frequency in the middle, the alpha, that's one of the key driving frequencies of the brain. It's the frequency in which consciousness is in part regulated. So you can see that Sentia has an impact to enhance alpha frequency, and that probably explains why it makes me more relaxed. And it also reduces, in a different part of the brain, it reduces gamma frequency, the high frequency, and that probably also reduces, is a measure of it reducing stress. And alcohol is different, and that's really important to us because we don't want to mimic alcohol because we don't want the problems of alcohol. We want our drinks to be different, and it is. Alcohol has different effects on different frequencies in different brain regions. And we're now in a process. We've just been awarded a BBSRC grant with Alex to take this field further forward over the next 3 years. So I mentioned that the world was interested in a new way of thinking about drinking, but most people weren't. Most people wanted to carry on drinking what they're drinking. So we decided to bite the bullet and make drinks that they wanted to drink rather than make them change their habits. So we've now taken the botanical components of Sentia, refined it, and put it into beers. So there's a stout and there's a IPA. So these are beers that have been made with alcohol. Alcohol is extracted, so they're alcohol-free, and you put in the botanical ingredient to get an effect. And we've also now launched Cask, which is a whiskey equivalent. And it's turned out to be rather popular. It's probably the first functional alcohol-free whiskey. There are plenty of other alcohol-free whiskies, but this is the first one that actually does something. And I want to make a special mention of Helen, who's here. She's there with the checkerboard on her. So she approached us a couple of years ago and said, look, I know quite a lot about how you analyze things. And we said, oh, good. I can help you work out what's in your botanicals. So with a master's student, she ran some of these HPLC traces for us to identify molecules that are already which were in the botanical ingredients. This particular one is, I think, passion flower, and so she's allowed us to look at the magnitude of the different elements, but also she's discovered some new molecules which we didn't know existed in that particular extract. No one had found them before. So we're building up a body of knowledge about what the actual key ingredients are in the botanicals that do what we want them to do to mimic alcohol. And maybe we will end up being able to refine some of those to make a more specifics, a small molecule, individual molecule, rather than a cocktail. And that would have some advantages because putting a lot of botanical ingredients into a drink is quite challenging in terms of, particularly in keeping it clear, which is very difficult. What about the synthetic? What about making the molecules that a single small molecule that you could put into drinks in the same way as Intel was the chip that went into computers. Our vision is that Alcarel will be the small molecule that we would license to any drinks company in the world, so they could make any drink they like that would be functional but not have alcohol in it. And we've got molecules. I showed you the function of one of them. We've got about 3 lead molecules. In fact, we're ready to go into humans. We've got them patented. The problem is, to get a new molecule into humans, you have to go through a pathway which is very similar to the pharmaceutical pathway. And that costs a lot of money. So I'm disappointed that Mr. Wharton left, because I was hoping that the regional government here might want to fund it, because we could certainly reduce quite a lot of problems in Liverpool and Manchester if people were drinking our drink rather than ethanol. But anyway, any of you rich, see me afterwards, please. But we're ready to go once we've got funding. And my own personal view is, I would have thought this surely should be an initiative that governments would really want to endorse. I mean, we're pumping billions into cleaner energy. Why don't we try to deal with the problem which costs us 30 billion? Alcohol costs the UK economy 30 billion a year. Would it be? All I'm asking for is 3 million. That's quite a good return, I would have thought. So I'm going to stop now, and I'll take questions. Thank you very much.
2 or 3 questions. I'm just going to do one now. We've got one here, one over there.
Hi, my name is Deborah. That was super interesting, thank you. My question is more in relation to health. So, one of the slides you showed looked at gut function.
Yeah.
And first I was thinking, why would you want to reduce gut function? Because obviously we need good gut function.
Yeah.
But I wondered if there's potentially any other uses for this product that you're creating other than social alcohol. Could it help with gut disease, things like that, irritable bowel disease, things like that?
Well, it could, yes. I mean, a lot of people with anxiety disorders we've all had butterflies in our stomach, haven't we? We've all had that kind of anxiety. A lot of people have chronic anxiety in their gut, so it could help with that. But also the other thing we're learning, which we didn't know about 2004 when that first paper was published, or the first book was published, we didn't even know there was a microbiome. Now we know there's a hell of a lot of different bugs in the microbiome, and many of them actually use GABA to promote growth and to and it's almost certain that the gut bugs releasing GABA are affecting your gut as well. So that's a whole new area we're beginning to explore, whether we can use our molecules to enhance gut health as well as relax people. Yeah.
Thank you very much. My name is Isabel. I'm from the Royal Society, and I think I have 2 questions. First is about GABA supplements, because I think they've been on the market for. I'm Russian, and in Russia we have so-called nootropic pharmaceutical supplements. Mm-hmm. And as a typical Russian, I'm used to think that this is not the real science, that there's because if you check those supplements, you will never find any research in English, and I was always thinking that this is something like snake oil, but your presentation is eye-opening for me. And I was just wondering, what do you think about it? What do you think about supplements? And another question was about, I think many people, I hear here and there that people stop drinking as much as they used to. Yeah. And I think empirically I feel the same, because I see that younger generations drink less and less. Yeah. And I was just wondering what's your take on this?
Well, I think they're smarter than the older generation, aren't they? That's who we're targeting. We want them to have a choice. In fact, it's interesting that the Sentia are sold to 2 groups. They're sold to older people who know that drinking's killing them and they want to drink less, and they're sold to young people who don't want to start. So we're actually surprisingly, our innovation has paralleled, or fortuitously been paralleled, by a social change in young people drinking less. In terms of the supplements, yes, so GABA itself doesn't get into the brain, it doesn't cross the blood-brain barrier, but there are GABA receptors in the gut. So GABA, if you take GABA in as a single molecule, you will almost certainly have some impact on GABA receptors in the gut, and you may very likely to change the microbiome by activating GABA receptors on some of the bugs. So it is quite plausible that GABA itself as a supplement will do things. It's not well established, but it is plausible. It's also very sweet and unpleasant, but apart from that, that's not. Some people like that. No, I don't.
You had me at no hangover when you were going through it. But serious question around if in the future we got rid of alcoholic drinks as we knew it, and we had alcoholic drinks that were actually based on this. How do we manage that? For example, now we've got a lot of legislation like driving, drink drive limits, drug drive limits. So how would we, if we were recreating some effects of alcohol, how would we then manage those with work, with driving and stuff like that?
Well, you'd have to use functional tests. I mean, of course, driving won't be a problem because Musk will be driving us everywhere in his car as well. So that's not a problem. One of the things what's really I didn't have time to explain in great detail, but our drinks are all targeted to have a plateau effect and not to exceed it. So you cannot get drunk, you won't fall over, you won't get addicted, you won't have hangovers. So if we were to regulate the future of drinking in a way which stipulated those criteria in the same way as we do, for instance, with the strength of things like paracetamol, we could almost eliminate all those problems. And you're from Wales, right? So I'm very happy to talk to the Welsh Government about brewing this stuff and making it in Wales. Right? Thank you.